Okay, so we're first going to talk about why Koch's postulates do not work on viruses. So Koch's postulates, if you'll remember, number one, you're going to find every case of the disease. Well, not every case, but in every case of the disease, you will find the same bacteria. So in every staph infection, you should be able to find Staphylococcus aureus, for example. So then you're going to isolate that bacteria and grow it in a culture. You're going to take the bacteria you grew in a culture and you're going to inject it into a healthy specimen, usually a mouse, and then that mouse is going to get sick. And then once you're done with that, you will isolate the bacteria from the mouse and grow it on a plate again. So that's how you prove that a certain bacteria causes a certain disease. So why would Koch's postulates not work on viruses? So what do we know about viruses? They are not living because they're acellular. They don't have the organelles and cellular processes like regular cells do, and they cannot reproduce on their own. They have to have a host cell. So you cannot grow them on a plate. They will not grow on a plate. So in Koch's postulates, you isolate the bacteria and grow it on a plate. Won't work. Viruses do not grow on their own. So today we're going to talk about pathogens. A pathogen is an agent which causes a disease. So why is this important? This is important because the germ theory states that disease is caused by microorganisms. So we've talked about pathogenic viruses. We've talked about pathogenic bacteria, and this is a pathogenic protist. This is trypsinoma. It's a blood parasite, and these are red blood cells. And then this is a pathogenic fungi. So this is a fly, and then out of its body is growing a fungus. So obviously the fly is dead, so the fungus killed it, now it's growing. So viruses and vaccines, we've already talked about a virus. So a virus is a box and genetic material. So that box can be a capsid, which is a protein coat, an envelope, or both. So bacteria do not have capsids. Capsids are just viruses. So it's just a box made out of proteins. That's all it is. So what is a vaccine? A vaccine is made from a weakened or killed form of the virus or bacteria. So you could have a vaccine for a virus or a bacteria. So it's going to be just a weak form or a dead form of the virus or bacteria, and you're going to inject that in yourself. And it allows, well, not yourself, you're going to let a trained professional do that, please. And your body is going to be able to fight that infection and gain immunity to it. So it's going to stimulate the body's immune system to recognize the agent as foreign, destroy it, and remember it so it can destroy it again. So when you get a flu vaccine, you're getting kind of the sad, decrepit little form of the flu so that your body can fight it easily and then be ready to attack, on a higher level when the actual flu vaccine or the flu virus gets you. So it destroys it faster than you can get the symptoms. So a vaccine, we want to remember, can prevent a virus and a bacteria infection. Antibiotics are drugs that can kill infectious organisms, so they can take care of everything except viruses. So not viruses, very important. So why not viruses? Viruses are not living, they do not have a cell wall, and cell walls are usually what antibiotics attack first. So if you take out the cell wall, the bacteria cell is dead. So many are naturally occurring chemicals, which come from soil-dwelling bacteria or fungus. So we're going to fight fire with fire, and we're going to kill bacteria and fungus with the chemicals we find inside of bacteria and fungi. So some bacteria will develop a drug resistance to antibiotics, and they can survive the antibiotic exposure, and this is called antibiotic resistance. So this is why sometimes you'll take antibiotics, your doctor will give them to you, and then you will still stay sick. You won't get better. So gram-positive versus gram-negative. Gram-positive had a lot of peptidoglycan in their cell walls. Gram-negative bacteria have a little bit of peptidoglycan in their walls. So gram-positive stained purple, gram-negative stained pink. So what I want you to gather from this slide is that cell walls differ between gram-negative and gram-positive. Therefore, the way antibiotics interact with these bacteria is going to be different. So antibiotics typically do not work against gram-negative bacteria because the cell wall is not exposed. So in gram-positive, it's just a cell wall. Then the antibiotics can attack it. In gram-negative, they have a cell wall, and they also have this outer membrane, and the bacteria can't get to the cell wall to destroy it. So vaccines versus antibiotics. Why do antibiotics not work on viral infections? So viruses are not living, they do not have a cell wall, so the antibiotic isn't even going to know it's there. It can't do anything to just a protein coat. So we talked about this already. Koch's postulates do not work on viruses because viruses cannot reproduce without a host cell. So you're not going to be able to grow them in culture, which is steps 2 and 4. So we've talked about other pathogenic bacteria. You don't need to know these. This is just stuff we're going to talk about. You do need to know biofilms. So Streptomyces, this is the cultured bacteria. Pharmaceuticals use this to produce antibiotics. And then chlamydia, this is, I'm sure you all know what chlamydia is. So Chlamydia trachomatis is a prokaryote which lives inside of eukaryotic cells, so it's parasitic. So biofilms, this is where prokaryotes, which are bacteria, different groups, different types, they gather together in a highly organized colony, and they cause urinary tract infections, ear infections, and they can gather on catheters and cause things like septic shock. So what's interesting about biofilms is they make this colony, and then whatever you take to get rid of it, it can't get to the middle. So it can't get to all the rest of this stuff in here. So many antibiotics target the ribosomes in bacteria, which are responsible for transcribing the proteins to make cell walls. So you have ribosomes, proteins, and these proteins make up the cell wall. And if you take out the ribosomes, you're not going to get the cell wall. So this means that the bacteria can't repair itself, and it can't make new cell wall for its new replicate. So it can copy its DNA, but it can't make a new bacteria cell. They can't reproduce. So the ribosomes in prokaryotes and eukaryotes are different. This is why we can take antibiotics and it leaves our eukaryotic ribosomes alone. It doesn't take out our ribosomes and kill us. Okay, so on these next few slides, you don't need to write anything. This is just so you understand that there are pathogenic protists and there are pathogenic fungi. So Plasmodium, this is a protist which causes malaria, so it's a pathogen. Pathogenic fungi, so of the 100,000 known species of fungi, 30% are parasites or pathogens. Dutch elm disease, it was a disease for the Dutch elms in England, and then they accidentally sent us over some logs that were infected, and it destroyed 70% of our Dutch elms. It was very, very sad. You can also have corn smut and ergots. These affect crops usually, and I have pictures on the next slide. So this is smut. Alright, and here's our normal corn. And actually corn smut is served as a delicacy in some countries. I personally would not be interested in eating that. These are ergots on rye, which is kind of like wheat. And this is going to infect your And if you eat ergots, you will get sick. And this is also the fungus that LSD is made out of, so you will hallucinate. It's not good. So anyway, don't eat ergots to try and hallucinate or anything. That's bad. Don't do that. Alright, so a lot of the food that you get at HEB, like your corn and tomatoes and squash, it's all been genetically modified, so we call them GMOs, genetically modified organisms. And these have all been genetically modified so much that it's pretty much the same plant over and over, especially corn. So each corn plant is pretty much identical. And if you have a whole crop of these corn plants that are the same plant, how easy is it to transfer this disease to each one? So this is very dangerous because it can just decimate an entire crop. Alright, so enclosure, biofilm, this is the gunky stuff that will build up from bacteria colonies, and it can cause urinary tract infections, ear infections, and it can form on a catheter and cause bad things like septic shock. So an antibiotic works by destroying the bacterial cell wall or destroying the ribosomes that will end up making the cell wall.